This video is on Unit 3.3, Passive Transport. So in transport, we're going to be talking about how the cell moves things in and out past its cell membrane, and I need to talk about what passive is as well. So if a person is really passive, that means they're kind of laid back, they're not very aggressive, they don't get in the middle of problems or conflicts. Passive transport is kind of related to that in that it doesn't require energy. So the cell doesn't need any energy to move things in and out of it through passive transport. Its second rule is that it always moves molecules from high to low concentration. A good example of this is whenever you're cooking something or baking something, let's say you're baking cookies in the oven. The smell is a molecule in the air. The odor molecules start at the kitchen and eventually spread out through the house. So they started at high concentration in the kitchen and they went to low concentration throughout the whole house. So they moved from an area of high concentration to an area of low concentration. Now let's talk a little bit about cells as examples. We've discussed three different types of passive transport. The first type is diffusion. Diffusion is just what we already talked about, movement from high concentration of an area to low concentration of that particular molecule. So the example I just gave was that of the molecule that makes cookies smell good. But it could also be molecules like food coloring. You put a drop of food coloring into water and it starts out very concentrated, but it eventually moves to lower concentrated areas. This action is called molecules moving with the concentration gradient. So moving from high to low concentration is considered with a concentration gradient. Just to show you an example of this, if we have a concentration gradient, that means we have a cell where there are things that are in the cell, maybe it's proteins or carbohydrates, in higher concentration than what's outside the cell. Or concentration gradient could be something that's inside the cell in lower concentration than outside the cell. What a concentration gradient is not is when molecules are in even amounts inside and outside of the cell. So this cell has these molecules in the same amount inside, 1, 2, 3, 4 molecules inside, as it does outside. So this is not a concentration gradient. These two, number 1 and number 2, are concentration gradients. And molecules will always move from high to low concentration. So in the first example, the molecules will move out because the inside of the cell has a higher number than the outside. In the second example, the outside is a higher amount, so they will move in. All right, in the cell, some examples of molecules that move in and out of the cell this way are any small molecules and any molecules that do not have a charge, so they don't have a positive or negative charge. Oxygen gas is a good example of this. This is like when we blew through the Q-tip models. We blew air through the models, and we showed that oxygen gas was small enough to get through the Q-tips. Okay, after just plain old diffusion, the next type of diffusion that cells can do is something called facilitated diffusion. And facilitated diffusion is when you have a helper. So if you facilitate something, that means you are helping that to happen. And in this case, we're helping diffusion to happen with the use of protein channels. So this was symbolized by the straws that went through your Q-tip models. So if you imagine again a cell, and in that cell membrane there are little channels that allow for certain molecules to get in and out of the membrane. And these would be molecules that wouldn't normally be able to get through. Examples of molecules that can't get through the membrane, can't get through the phospholipid part of the membrane, are ions because they have a plus or a minus charge. and glucose because it's a little too big. So glucose and ions can go in and out through these channels. So imagine I have lots of glucose molecules inside this cell. Maybe these glucose molecules have been made by the cell, like a plant cell makes glucose. It will leave the cell through these protein channels because it can't fit through the phospholipids. The opposite could happen as well. Say I have some ions on the outside of the cell that need to get in. They can't go through the phospholipids, so instead they're in high concentration on the outside, so they're going to move in to the cell through these different protein channels. That's called facilitated diffusion. Diffusion, still high to low concentration, but with the help of protein channels. All right, the last type of passive transport that you need to learn for class is called osmosis. And osmosis is specific in that we are moving water across a membrane. So this basically happens most of the time in cells, and it's only the movement of water. It's still high concentration to low concentration of water, but it's very specific to water. Before we get into how this works, I need to define a couple words for you that you learned in 9th grade science but maybe need a reminder. When you have a solution, which is a mixture of a solvent and a solute, they always have these two parts. The solvent is the dissolver, and the solute is what's being dissolved. So if you were to make a pitcher of Kool-Aid, the solvent would be water, and the solute would be the Kool-Aid. Now this comes into play with osmosis because the amount of water that's in a solution is dependent on how much solute there is. So if you have a lot of solute, then you have less solvent, and vice versa. If you have a lot of solvent, then you have less solute, comparatively. So we're going to look at how osmosis works with moving water from high concentration to low concentration.